Introduction. 3 types of asexual reproduction. Growth process in which an organism gives rise to offsprings similar to itself. Reproduction. Biological process ensuring continuity of species on Earth. Generation after generation. Organisms' habitat mechanism depends on diversity in structure. Death. Lifespan not linked to size of organism. Internal physiology. A. Fission. B. Fragmentations. Budding process in which in the body of the organism individual forms 2 or more breaks into distinct pieces, daughter individuals types fragments, and each fragment grows into an adult capable of a. External budding producing offsprings. A small bulge, unequal division, from the parent body that develops externally and eventually separates from the example, Hydra types. Parameter 1. Binary fission. 2. Multiple fission. Parent operational conditions. Favorable and example, Hydron. Favorable. 5. Multiple. No number of daughters. Spindle formation. 2. Yes. Mouth. Example, right pointing arrow. Amoeba. Environmental factors. Organism. Butterfly, crow, tortoise, parrot. Lifespan 1 to 2 weeks, 100 to 150 years, 140 years. 15-year life cycle of an organism. No individual is immortal except single-celled organisms. 2 Types of reproduction. Parameters. Asexual. Sexual. Number of parents. Uniparental. 1. Uniparental. Tenure or 2. Biparental. Periplaneta mechanism. Yes. Gamete formation. Yes. Right pointing arrow. Gamete fusion. O. Type of cell division. Mitotic conditions for occurrence mostly favorable, identical to parent Hydra. Planaria nature of offsprings. Examples. Withdrawal of pseudopodia bud. Yes. Meiotic and mitotic both favorable and unfavorable, mostly non-identical offsprings. Ferritimer nucleus. Encystation. 3-layered cyst. 2. Internal budding advantages. Simple process. Contributes to evolution significantly through genetic recombination. Multiple fission. Excitation. Black circle. Internal buds called game moles that are aggregates of archaeocytes protected by spicules. Black circle. Rapid, faster. Black circle. High adaptability. Elaborate disadvantages. Expensive. No contribution to black circle. Complex evolution as genetic recombination. Crossing over is absent. Example, sponges. Daughter cells. Sporulation. Low adaptability. Black circle. Slower process. Parthenogenesis. Black circle clone. Morphologically and genetically similar individuals produced asexually. Black circle cell division is in itself a mode of reproduction in unicellular organisms, e.g., amoeba. Paramecium. Absence of gamete fusion. Syngamy. Female gamete develops into new organism without fertilization. Examples: rotifers, honey bees, some lizards, birds, turkey. A game mole. Types of gametes. Bases. Morphological and physiological difference. Sexual reproduction. Remarkable fundamental similarity occur in process of sexual reproduction among different organisms, despite different structures for reproduction. 2. Reproductive processes and associated behaviors are regulated by hormones and environmental factors. Heterogametes greater than different homogametes. Isogametes. Same events. Associated gametogenesis. A. Pre-fertilization events. Types. B. Gamete transfer. Enables bringing together of an gametes physically leading to fertilization. Gamete transfer. Parameters. Female malia. Gametogenesis. Formation of gametes. Size. Big small melanocytes. Gamete forming diploid cells. Gamete mother cells. Right pointing arrow. Gametes are haploid in nature, i.e. receive only one set of chromosomes at the end of meiosis. Motility chromosome number same internal fertilization external fertilization right pointing arrow a haploid parent produces gametes by mitotic divisions right pointing arrow A. Diploid parent produces haploid gametes by meiosis. Fertilization occurs inside black circle. Fertilization occurs in water. Food storage number reproductive tract less more black circle mostly terrestrial organisms, e.g. reptiles, mammals mostly aquatic organisms. Name of organism chromosome number in myocyte 2n, chromosome number in gamete n, e.g. bony fishes and amphibians. Ovum humans 23 disadvantages pre-reproductive phase juvenile 46 housefly 126 right pointing arrow limited number of progeny sperm types of organisms rat 4221 offsprings are extreme vulnerable to predators threatening their survival. B. Adulthood dogs 7839 unisexual bisexual hermaphrodite cat 3819 leech cockroach fruit fly puberty 84 great synchrony required between enduring external fertilization enhance chances of fertilization. Heterogametes are present in majority of sexually reproducing organisms. Butterfly 380 earthworm spongers 190 breeding season basis. The time duration when mating occurs reproductive phase. B. Fertilization zygote most vital event of sexual reproduction is fusion of gametes called syngamy resulting in formation of diploid. Single-celled structure vital connecting link ensuring continuity of species between generations. C. Post-fertilization, embryogenesis. Process of development of embryo from the zygote through mitotic cell divisions help increase number of cells and cell differentiation helps in formation of specialized tissues and organs. Location of development of zygote. Oviparous animals. Viviparous animals inside the body outside the body. End of reproductive phase is a protected by calcareous shell absent lesser parental care parameter of senescence slowing of metabolism greater parental care ultimately low adds to death. The chances of survival of young ones is greater in viviparous organisms. Parameters continuous breeders seasonal breeders breeding time examples throughout the year primates hens poultry birds in captivity specific breeding elephant birds in with horse cow goat 7 reproductive cycles in placental mammal cyclical changes during reproduction post-reproductive phase death parameters estrus cycle menstrual cycle greater than characteristic of non-primate mammals primates humans apes monkey examples dogs tiger cows deer sheep rats introduction accessory ducts primary sex organs site for gamete formation facilitate transport of gametes. Humans are sexually reproducing viviparous organisms. External genitalia involved in copulation. Reproductive system is composed of parts of fallopian tube The male reproductive system 3 female reproductive system uterine fundus last part of oviduct narrow lumen ureter location pelvic region uterine cavity isthmus location pelvic region accessory glands urinary bladder joins the uterus oviduct fallopian tube 10 to 12 cm length seminal plasma seminal vesicle vas deferens amper wide apart from these one pair extend from ovary infundibulum funnel shaped urethra contains fructose calcium enzymes, prostate gland to uterus, 1. Fibri finger-like epididymis. Uterus, Womb ovary projections that collect ovum after its secretions lubricate the penis bulbothral vasa efferentia rete testis inverted pear-shaped cervix cervical canal gland, 1 pair, attached to pelvic wall by birth ovulation ligaments vas deferens receives a duct from seminal vesicle and testicular lobules canal vagina opens into the urethra as the ejaculatory duct test is glans. Penis foreskin urethral meatus uterine wall consist of 3 layers. External genitalia of male, penis, 1. Endometrium lines lumen, glandular and undergoes cyclic changes during menstruation 2. Myometrium thick layer of smooth muscles that show strong contractions during delivery parts features 3. Perimetrium external thin membrane urethra originates from the urinary bladder and extends through the penis external genitalia of female. Special tissues help in erection of penis to facilitate insemination parts features glans penis enlarged end of penis covered by loose fold of skin called foreskin mons pubis cushion of fatty tissue covered by skin and pubic hair labia majora fleshy folds of tissue that extend down from mons pubis and surround the vaginal opening primary sex organs labia minora paired folds of tissue under the labia majora parameter male-female organ. Number test is ovary clitoris tiny finger-like structure which lies at the upper junction of labia minora above the urethral opening 22 shape oval almond hymen membrane that partially covers the opening of vagina location outside abdominal cavity in a pouch called scrotum lower abdomen. One on each side black circle can be torn while sudden jolt, fall, horse riding, cycling, insertion of vaginal tampon may or may not be torn during the first coitus, so its presence or absence is not reliable dimensions indicator of virginity or sexual experience. Length 4 to 5 cm. Width 2 to 3 cm. Length 2 to 4 cm covering dense connective tissue. Outermost, thin epithelium. Outermost, functions sperm formation, synthesize steroidal testicular hormones like androgens over formation. Synthesize steroidal ovarian hormones like estrogen and progesterone compartments 250 testicular lobules peripheral cortex and inner medulla zones in ovarian stroma have follicles in various developing stages. Scrotum helps in maintaining the temperature 2 to 2.5 degrees Celsius lower than body temperature. Necessary for spermatogenesis. 1 to 3 coiled seminiferous tubules. Lobule interstitial spaces outside seminiferous tubules contain immunocompetent cells and Leydig cells lining the seminiferous tubules. Functions 1. Male germ cells. Spermatogonia. Sperm formation. Ovary is connected to pelvic wall and uterus by ligaments. 2. Sertoli cells provide nutrition to the germ cells. Primary blood follicle. Tertiary follicle showing antrum fig. Sectional view of ovary agametogenesis process of gamete formation. Spermatogonium. Parameters male-female oogenesis. Sertoli cell term spermatogenesis at puberty. Primary spermatocyte process begins during embryonic development. Ploidian chromosome in fetus number per cell 2 n equals 46 corpus female mother cells, arrested at prophase I, secondary. Spermatocyte male germ cells. Spermatogonia. Oogonia mitosis and differentiation. Spermatozoa follicles. Characteristics 2 n equals 46 primary spermatocytes. Primary oocytes. Primary single layer of granulosa cells. Periodic meiosis I. Birth fig. Sectional view of seminiferous tubules. More layers of granulosa cells and new theca layer. Puberty. Secondary semen equals 23. Completion of meiosis I. Reduction division. Secondary spermatocytes 1 polar body secondary oocyte tertiary secretions of epididymis and vas deferens are essential for maturation and motility of sperms. Fluid-filled cavity antrum and the layers are organized into external and internal layers. Meiosis II. Male ejaculates about 200–300 million sperms during a coituscence. Equals 23 spermatids graphian. Mature secondary oocyte forms a cellular zona pellucida around it for normal fertility. Spermiogenesis 60% sperms must have normal shape and size. 40% of 60% sperms must show vigoration equals 23 sperms 2 polar body ovum ovulation motility spermiation nomo-oogonia are formed and added after released from seminiferous tubules birth during the embryonic development. A couple of million gamete mother cells, cogonia, are formed within each fetal ovary. Hormonal regulation in males2 n diploid n equals haploid right pointing arrow. A large number of follicles degenerate from birth to puberty, so only 60,000 to 80,000 primary follicles are left in each ovary at puberty. Meiosis in oogenesis results in unequal-sized cells and the secondary oocyte retains bulk of the nutrient-rich cytoplasm of the primary oocyte. Fate of polar body is not certain. Hypothalamus secretes GnRH. Significant amount. Sperm targets anterior pituitary main parts secretes acrosome cap like gonadotropins. Spermiogenesis is structure filled with enzymes help in fertilization transformation of spermatids to sperms and sperm head is embedded in Sertoli cells head seminiferous tubules outside 8 structure of gametes nucleus n elongated FSH-LH neck ovum right pointing arrow plasma membrane acts on middle piece. Mitochondria provide energy source for swimming, movement of tail. Sertoli cells lay egg. Interstitial cells synthesize and release the function of male sex. Accessory ducts and glands are maintained by the testicular hormones. Androgens, cells of the facilitate sperm motility which is essential for fertilization. Some factors: androgens, testicular hormones. Zona plus pellucida corona radiata perivitallin. Space tail stimulates spermiogenesis. Spermatogenesis hormonal regulation in females and menstrual cycle. The cycle of events starting from one menstruation till the next one is termed menstrual cycle. Characteristic of female primates. Monkeys. Menstrual hygiene 1. Maintenance of hygiene and sanitation during menstruation is a very important. Cycle occurs if ovum remains unfertilized. Humans too. Take bath and clean yourself regularly. Use sanitary begins at puberty. Menarche. Lack of cycle may be an indication of reproductive phase. Napkins. Clean homemade pads ceases around 50 years. Menopause. Pregnancy. Stress. Poor health etc. Point 3. Change sanitary pads after every 4 to 5 hours. Right pointing arrow. Average duration in humans equals 28–29 days. 4. Dispose of used sanitary napkins properly by wrapping it in menstrual cycle used paper. Changes in the ovary and the uterus are induced by changes in the levels of pituitary and ovarian hormones. 5. After handling the napkin, wash hands with soap. Phase duration hormones and their effects. Events in ovary. Events in uterus. Menstrual 3 to 5 days. Drastic decline in progesterone. Corpus luteum degenerates. Black circle breakdown. of endometrial lining and its blood vessels, which forms liquid that comes out through vagina, constituting developing corpus luteum, regressing corpus luteum, developing follicle, mature follicular flow, LH surge, FSH follicular variable ovulation or proliferative phase, reproductive cycle, primary follicle gradually matures to Graafian follicle, gradual increase in FSH and LH that stimulate secretion of estrogen from follicles, endometrium regenerates through proliferation, estrogen, estrogen peak, progesterone, ovulation 14-day, middle of cycle, FSH and LH at peak. LH surge, rupture of Graafian follicle and release of only one ovum. Cycle proliferation of endometrium continues. Menses luteal secretory fixed, 14 days, secretion of progesterone and estrogen, remnants of the Graafian follicle transforms into corpus luteum, days 3-5, menstruation. Endometrium is maintained, if ovum remains infertilized, endometrium is sloughed off, marking a new cycle. 9-1-1-1-3 follicular phase, proliferative phase, 1-5-1-7, 1-9, 2-1, 2-3, 2-5, 2-7, 2-9, 1 luteal phase. Next cycle, secretory phase, begins if ovum gets fertilized. Endometrium is maintained by progesterone necessary for implantation and other events of pregnancy. During pregnancy, all events of menstrual cycle stop. Development of the zygote, down pointing arrow, allow every sexually reproducing organism, including human beings, begin life as a single cell, the zygote. The process of development of embryo from zygote is called embryogenesis. During embryogenesis, zygote undergoes cell divisions and differentiation. Cleavage starts as zygote moves through isthmus to the uterus. Daughters formed after cleavage are called blastomeres. 2 cells, 4 cells. Introduction changes in gametes during fertilization, secretions of acrosome, part of sperm. Sequence of reproductive events occurring in humans include sperm gametogenesis, entry of sperm into cytoplasm of oocyte through zona pellucida and plasma membrane cells of the corona radiata. Zona pellucida, perivitelline space, insemination, fertilization. Changes in zona pellucida prevent entry of additional sperms and ensures that only one sperm can fertilize an ovum. Implantation, fig ovum surrounded by few sperms. Gestation, parturition, birth inducing completion of meiosis 2 of secondary oocyte form. Cytoplasmically unequal 2 inches. Meiotic division 2 path followed by gametes in female. Reproductive T. RACT2 polar body, N. Ovum, oocyte, N. 1. Sperm, N. During coitus, semen is released from male reproductive tract by the penis into the female reproductive tract. The vagina by process termed insemination. Nuclei fuse, released in perivitelline space, zygote, 2N. Sperms, oocyte released in body cavity, released in vagina. Characteristics, swim through vital link that ensures continuity of species between organism of one generation and the next. Captured by black circle. Sex of a child is decided at this stage. Sex of a baby is determined by the father. Cervix, fibrillantous uterine fundibulum. Reach parameters: female XX, male XY. Chromosome pattern: AMPA. Site of fertilization: gametes formed 1 cell. Zygote 8 to 16 cells. Morula, trophoblast, outer layer of blastomeres attaches to endometrium. Inner cell mass, Inner group of cells attached to trophoblast. Right pointing arrow differentiates into embryo with 3 germ layers. Blastocyst implants in uterus. Fusion of gametes, syngamy, fertilization, vital event of sexual reproduction. The inner cell mass contains certain cells called stem cells which have the potency to give rise to all the tissues and organs. After attachment, uterine cells divide rapidly and cover the blastocyst. Embedding of blastocyst in endometrium is called implantation. Leads to down pointing arrow. Pregnancy. Fusion of gametes. Female XY male. Fertilization can only occur if the ovum and sperms are transported simultaneously to the ampullary region. This is the reason why not all copulations leads to fertilization and pregnancy. 50% male gametes carry X chromosome and 50% carry Y chromosome. Zygote would carry either XX or XY depending on whether the sperm carrying X or Y fertilizes the ovum. Germ layers. Outer ectoderm, middle mesoderm, endoderm, inner. These together give rise to all tissues, organ in adults. Gestation. Periodcanges. After implantation, embryo, fetus, mother finger-like projections appear on the trophoblast called coronach villi. Average duration of pregnancy in dog 63 days, cat 63 days, elephant, human 18 to 22 months. 9 months. Uterine tissue. Major events during gestation period in humans. Trimester, month, week. Event 4. Interdigitate to form structural and functional unit between embryo, fetus and mother. Placenta. Supply of O and nutrients to the embryo. Removal of CO and excretory waste materials produced by the heart is formed. Sign of growing fetus noticed by listening to the heart sounds through stethoscope. Functions. B. Fetus develops limbs and digits. E and 12. Most of major organ systems are formed including external genital organs. Embryo acts as endocrine tissue. V. Produced only during pregnancy. 20 hcg. Human coronach gonadotropin. First movement of fetus. Appearance of hair on head. 2 V and 24 hpl. Human placental lactogen body is covered with fine hair. Eyelids separate. Eyelashes are formed. IX and 36. Fetus is fully developed and is ready for delivery. Relaxin, also secreted by ovary in later phase of pregnancy. 3C. Estrogens increase several folds during progestogens. Pregnancy. Essential for supporting PARTURITION. Cortisol. Fetal growth. Placenta is connected to the embryo through an umbilical cord which helps in the transport of substances to defined as delivery of the fetus, childbirth, metabolic changes in mother. Prolactin signals for parturition originate from fully developed fetus. Maintenance of pregnancy. Thyroxine. Placenta. Placental villi from the embryo lead to cavity of uterus, mammary glands and lactation. Mild uterine contractions. Functional mammary gland is characteristic of all female mammals. Paired structures, breasts, that contain variable amount of fat and called yolk sac. Umbilical cord. Glandular tissue with its vessels. This is positive fetal ejection reflex. Embryo divided into feedback loop triggers release of 15 to 20 mammary lobes. Oxytocin from posterior pituitary of mother contain causes mammary alveolus, plug of mucus in cervix. Cluster of cells. Strong uterine contractions. The human fetus within the uterus secrete and store milk. In their lumen oxytocin is rebleeding to areola synthesized by opens into muscles between ribs. Its source gland. Hypothalamus. Expulsion of baby through birth canal. Parturition, nipple. Childbirth is induced by a complex neuroendocrine mechanism involving cortisol. estrogens and oxytocin. Doctors inject oxytocin to induce delivery. Mammary tubules but released from posterior pituitary, join to form followed by pectoralis major muscle mammary duct expulsion of placenta many join to form undergo. Differentiation during pregnancy. Mammary amper black circle mammary glands secrete milk after childbirth that helps mother in feeding newborn by process called lactation, connected to fig. Sectional view of mammary gland. Milk produced during initial few days of lactation is called colostrum, which contains several antibodies. Absolutely essential to develop resistance for the newborn babies. Leptiferous duct, through which milk is sucked out. Breastfeeding during the initial period of infant growth is recommended by doctors for bringing up a healthy baby. Introduction. Definition according to WHO. Reproductive health means a total well being in all aspects of reproduction, i.e. physical, emotional, social and behavioral. Point 2. Reproductive health. Problems and strategies. India was amongst the first countries in the world to initiate action plans to attain total reproductive health such as family planning programs, FPP, in 1951, helping adolescents by providing information about reproductive organs, hygienic sexual practices and STIs, importance of breastfeeding, creating awareness about care of pregnant mother, postnatal care of mother and child. 3. Population stabilization. According to 2011 census report, our population growth rate is less than 2%, i.e. 21,000. Year world population. Indian population 1900 2,350,000,000 2006 billion 1 billion. Awareness regarding social evils such as sex abuse and sex-related crimes, discouraging children from believing in myths and having misconceptions about sex-related aspects. 2011 7. 2 billion 1.2 billion. Importance of equal opportunities for and child tasks performed by reproductive and child health care (RCH) programs. Role of NGOs, audiovisual and print media in creating awareness regarding sex-related aspects, educating people about birth control options, benefits of massive child immunization, reasons for increase in population size, decline in death rate, rapid decline in maternal mortality rate, MMR, black circle decrease in infant mortality rate, IMR. Increase in number of people in reproducible age, black circle increase in health facilities, measures taken by government to check population growth rate, motivate smaller families by using various contraceptive methods with slogans Hum do Hamare do, advertisements and posters, urban couples adopting one-child norm, statutory raising of marriageable age, black circle female to 18 years, black circle male to 21 years, incentives given to couples with small families, benefits of families with small size, introduction of sex education in schools, artificial methods. 5 methods work on the principle of avoiding physical meeting of the egg and sperm's chances of failure are high. The various methods of contraception are enlisted below. Birth control, contraception features of an ideal contraceptive, user-friendly, easily available, effective, reversible, no least side effects, no interference with libido and act of coitus. There are 2 principal methods of birth control: natural methods, artificial methods, barrier methods. Method, mode of action, Moya, I. Spermicidal jellies, periodic abstinence, couples abstain from coitus from day 10 to 17. 3. IUDSOF, the menstrual cycle, a fertile period. Withdrawal method, coitus interruptus. Insemination is avoided as the male partner IV. Oral pills withdraws his penis from the vagina just prior to ejaculation v. Injections and implants VI. Emergency contraceptives black circle lactational amenorrhea, absence of menstruation up to 6 months during period of intense lactation following parturition VIA. Surgical methods artificial methods IV. Oral contraceptive pills, OCP, or tablets VIA. Surgical, sterilization methods or poor reversibility but highly effective parameter non-steroidal steroidal mode of action example or Saheli, condoms and its types progestogens, PRG, alone blocks gamete. Transport types made up of rubber or thin latex less than composition or combination of praga and estrogens, est, inhibit ovulation and males-females mode of action interferes with tubectomy vasectomy penis black circle vagina and cervix implantation. Also black circle in females black circle in males yes alter the quality of cervical mucus to retard. Cut and tie fallopian black circle cut and tie vas tubes deferens entry of sperm small incision on the scrotum dosage on say a week. Pills have to be taken daily for a period of 21 days starting preferably within first 5 days of menstrual cycle male condom, Nirod, female condom 8 medical termination of pregnancy, MTP, induced abortion, B, diaphragms. Cervical caps, walls. Rubber barriers that cover the cervix during coitus effectiveness high contraceptive value with very few side effects. Pills are very effective with lesser side effects and well accepted by females. MTP. Intentional or voluntary termination of pregnancy before full term. Reusable. Do not protect from STIs. Used by females only. MTP was legalized in India in 1971. When can MTP be performed? Saheli was developed at CDRI, Lucknow, Uttar Pradesh. 1. Spermicidal jellies. Foams and creams unwanted pregnancy due to rape, failure of contraception, casual unprotected intercourse. Kill the sperms by creating acidic pH. Used along with barrier methods to increase their efficiency. V. Implants. IF continuation of pregnancy could harm the mother or fetus or both. I. Intrauterine devices, IUDs, placed under skin inserted by doctors or expert nurses in uterus. IUDs are one of the most widely accepted method of contraception in effective periods are much longer. Permission of how many medical practitioners is needed for MTP depending on duration of gestation. India. Composition. Progestogens alone. Combination of progestogens and estrogens types black circle mode of action. Moya. Non-medicated IUDs. Medicated IUDs black circle inhibit OV. Ovulation and implantation alter the quality of cervical mucus. Example. Lips lupus then, up to 12 weeks more than 12 but less than 24 weeks to retard entry of sperms parameter. Examples mode of action black circle injections usually share similar moya and composition as implants. Intention behind MTP Amendment Act 2017. Hormone-releasing IUDs progestazate. LNG20 copper-coated IUD SCU7, multiload 375, goal, make the uterus unsuitable for implantation and cervix hostile to the sperms, increase phagocytosis of sperms within uterus via emergency contraceptives. Black circle reducing the incidence of illegal abortion. Co-ions suppress sperm motility and fertilizing. Black circle decrease consequent maternal mortality and morbidity. Types, characteristics, capacity of sperms. Progestogens alone, effective within 72 hours of coitus, used to prevent conception. Black circle MTPs are safe up to 12 weeks but riskier in second trimester, yet both are legal. Combination of PRG plus est resulting from rape or unprotected intercourse. Amniocentesis and MTPs have been misused in context of female feticide. Amniocentesis 11 infertility. Analyze fetal cells and dissolved substances from infertile couple unable to produce children in spite of 2 years of unprotected sexual cohabitation. Amniotic fluids technique used to check for genetic disorders such reasons for infertility as Down syndrome, hemophilia, sickle cell anemia etc. Physical. Congenital infertility is a problem could be with either the male or female partner. In India, female is blamed often than male for the couple being childless. Diseases. Immunological statutory ban on this technique in India to prevent female feticide. Psychological help for infertile couples comes in the form of assisted reproductive technologies, ART, sexually transmitted infections, STIs, other details of ART involved, alternately named venereal diseases, VD, or reproductive tract infections, RTIs, parameter in vitro fertilization, in vivo fertilization outside the body in simulated, in the female ICSI intracytoplasmic sperm injection. Site of fertilization. High vulnerability, risk group, 15 to 24 years. Conditions in laboratory. Black circle sperm injected directly into the egg. Artificial insemination, AL. Reproductive tract. No, yes. Can female? Yes. Mode of transmission, MOT. Sexual intercourse produce over semen introduced in vagina or uterus. Black circle embryo transfer. Yes. Category disease, low sperm count or inability of male to inseminate female. Bacterial. Example of gift, AI, G. Black circle ZIFT, IUT, ICSI. Gonorrhea, syphilis. Chlamydiasis techniques. Protozoan trichomoniasis. Black circle IUI, intrauterine insemination. Viral genital herpes, hepatitis B, genital warts, AIDS. GIFT. Gamete intrafallopian transfer. Site of embryo transfer, ET, based on number of blastomeres. Black circle female can provide conditions for fertilization and further development. Bacterial and protozoan diseases are completely curable if detected early and treated properly. Parameter up to 8 blastomeres. More than 8 blastomeres. Other MOT for hepatitis B virus and HIV infection include locationing fallopian tube in uterus technique. ZIFT. Zygote intra-IUT, intrauterine transfer test tube baby program involves TESHIX with in vitro fertilization. Sharing of injection needles. Surgical instruments with infected persons. Fallopian transfer. No transfusion of blood. Right pointing arrow. Symptoms and complications of STIS from an infected mother to fetus. Early detection. Symptoms. Late detection. Complications. Preventive measures to avoid STIs. Avoid sex with unknown partners. Multiple partners. Always try to use condoms during coitus. Itching, fluid discharge, pelvic inflammatory diseases, PIDs, slight pain, swellings in the genital region. Abortions, stillbirths, ectopic pregnancies, infertility. Cancer of reproductive tract evolution. The ORIES for Origin of Life. Evolutionary biology is the study of history of life forms on Earth. Stellar distances are measured in light years. Big Bang explosion. Singular huge explosion. The origin of life is considered a unique event in the history of universe theory. Proponents' connotations resulted in leading to origin of Earth. Special creation. Conventional religious literature. All living organisms that we see today were created as such. Parameters. Origin of universe. Time scale. 20 billion years ago. Black circle. 4.5 billion years ago occurred in solar system of Milky Way galaxy. Black circle comprises cluster of galaxies, stars, clouds of gas, dust. Diversity was always the same since creation and will be the same in future also. Feature. Earth is 4,000 years old. Events after expansion of universe. Cosmozoic, panspermia. Black circle gases condensed. Early Greek thinkers, astronomers. Black circle temperature declined. He formed life came from outer space. Galaxies of present-day formed units of life called spores were transferred to different planets including Earth. Hypothesis for origin of life on early Earth. No atmosphere existed on early Earth. Water vapors, methane, carbon dioxide and ammonia released from spontaneous life came out from decaying and rotting matter generation like straw, mud, etc. Disapproved by Louis Pasteur. Molten mass covered the surface. Louis Pasteur, New Hampshire 37, H. UV rays H plus O2H plus 0 plus CO2 plus H2O plus other contents. Hydrogen oxide vapor falls as rain to fill all the depressions and form oceans. Theory of life comes only from pre-existing life. Biogenesis. He showed that in pre-sterilized flasks, life. Ozone layer was formed did not come from killed yeast, Achillidae yeast. Pre-sterilized flasks. No life. Oparin Haldane hypothesis or chemical Oparin. Russia. Haldane, England. Origin of life. Life appeared 500 million years after the formation of Earth, i.e. almost 4 billion years ago. Probably originated 3 billion years ago. Electrodes. Evolution. Spark discharge. Simulate lightning, energy source. First non-cellular forms of life would have possibly originated from giant molecules, RNA, protein, etc. And at black circle formation of life was preceded by biochemical evolution, i.e. formation of diverse organic molecules from inorganic constituents. First form of life could have come from pre-existing non-living organic molecules, e.g. RNA, proteins, etc. This hypothesis was proved by Miller's experiment, 1953, S. L. Miller, American scientist. In similar experiments others observed formation of sugars, nitrogen bases, pigments and fats. Analysis of meteorite content also revealed similar compounds indicating that similar processes are occurring elsewhere in space. 800 degrees C. These capsules reproduced their molecules. Perhaps vacuum simulate reducing atmosphere. Closed task possibly originated 2 billion years ago. First cellular forms of life arose in water environment, aquatic, boiling water. Amino acids. This version of biogenesis, i.e. the first form of life arose slowly through evolutionary forces from non-living molecules is accepted by majority. Fig. Diagrammatic representation of Miller's experiment. Evidences of evolution. 1. Paleontological evidences. Evidences from fossils, 3. Morphological and anatomical evidences. Black circle fossils are remains of hard parts of life forms found in rocks. Parameters. Common ancestry. Anatomical structures. Black circle age of fossils is determined by radioactive dating method. Black circle fossils of different life forms in different sedimentary layers indicates the geological period in which they existed. Epochs. Periods. Eras. Black circle rocks form sediments and a cross-section of Earth's crust indicates the arrangement of sediments one over the other during the long history of Earth. Black circle different aged rock sediments contain fossils of different life forms who probably died during the formation of the particular sediment. Some of them appear similar to modern organisms. They represent extinct organisms, e.g., dinosaurs. 2. Embryological evidences proposed by Ernst Haeckel. Based upon the observations of certain features during embryonic stage common to all vertebrates that are absent in adults, e.g., embryos of all vertebrates develop a row of vestigial gill slits functional only in fish and not found in another adult vertebrates. It was disproved by Carl Ernst von Baer. He noted that embryos never pass through the adult stages of other animals' homologous organs, analogous organs, similar but developed along different directions due to adaptations to different needs. Function perform type of evolution examples different divergent vertebrate hearts. Vertebrate brains molecular homology. Thorn of bougainvillea and tendril of cucurbita lemur JX not similar but resulted in selection of similar adaptive features in different groups of organisms, thus evolving for the same function. Similar convergent placental mammals Australian marsupials thorn anteater numbat, anteater spotted cus forelimbs of vertebrates bobcat Tasmanian tiger. Cat wings of butterfly and bird's eyes of octopus and mammals flippers of penguins and dolphins black circle sweet potato, root modification, and potato, stem modification, similarities in proteins and genes. Performing a given function among diverse organisms indicates common ancestry. These biochemical similarities point to the same shared ancestry as structural similarities among diverse organisms. Adaptive radiation theories for the evolution of life forms. The process of evolution of different species in a given geographical area starting from a point and literally radiating to other areas of geography, habitats. Examples Lamarck's theory of evolution black circle given by French naturalist Lamarck use and disuse of organs inheritance of acquired characters main postulates, Australian marsupials Darwin's finches. Small black birds evolved from seed-eating birds in Galapagos Island altered beaks arose, enabling them to become insectivorous and vegetarian finches. Tasmanian wolf sugar glider marsupial mole tiger cat black circle those characteristics which enable some to survive better in natural conditions. Climate, food, physical factors etc. would outbreed others that are less endowed to survive under such natural conditions. The fitness, according to Darwin, refers ultimately and only to reproductive fitness. Adaptive ability is inherited and has a genetic basis. Black circle fitness is the end result of the ability to adapt and get selected by nature. Black circle branching, descent and natural selection are the two key cons. PTS of Darwinian theory of evolution. Habitat fragmentation and genetic drift may accentuate variations leading to speciation. Example: Long neck in giraffes is attributed to stretching the neck while foraging leaves on tall trees. Darwin's theory of natural selection. Darwin visited Galapagos Island. Ship HMS Beagle. Observations by Charles Darwin. Sad face. Existing living forms share similarities to varying degrees not only among themselves but also with life forms that existed millions of years ago. The geological history of Earth correlates with the biological history of Earth. Influence of the work of Thomas Malthus on marsupial radiation. Banded anteater, koala, bandicoot, marsupial rat, wombat, kangaroo, fig. Adaptive radiation of marsupials of Australia. Fig. Variety of beaks of finches that Darwin found in Galapagos Island when more than one adaptive radiation appeared to have occurred in an isolated geographical area, representing different habitats. One can call this convergent evolution. Darwinism. Weaknesses of Darwinism. Examples: placental mammals, Australian marsupials, mole. Population can grow exponentially. Limited resources. Nature keeps control. Check on population size, which are hence nearly stable. Darwin's postulates. According to Darwin, variations are small and directional, and evolution for Darwin was gradual process. Anteater, marsupial, mole, numbered. Banded anteater, marsupial, mouse, mouse. 1. Unable to explain the origin of variations. 2. Could not explain speciation. Ignored work done by Mendel. Mutation theory given by Hugo de Vries in first decade of 20th century. Worked on evening primrose. New species originate as a result of mutations which are single-step. Saltation. Large, random and directionless variations arising suddenly in a population. Lemur, spotted cuscus, flying squirrel, flying phalanger, sugar glider, Tasmanian tiger, cat, bobcat, black circle. Darwin asserted that in a population exist variations which are heritable and which make resource utilization better for few, will enable only those to reproduce and leave more progeny. Wolf, Tasmanian wolf. Biological evolution. Metabolic capabilities of different cellular forms of life under the influence of natural selection contributed to biological evolution. Rate of appearance of new forms is linked to the life cycle or the lifespan. Microbes have the ability to multiply and become millions within hours, whereas fish or fowl would take millions of years as lifespan of these. Black circle. Alfred Wallace, a naturalist who worked in Malay archipelago, had also come to similar conclusions as Charles Darwin around the same time. When we describe story of this world, we describe evolution as a process. When we describe the story of life on Earth, we treat evolution as a consequence of a process called natural selection. Black circle. Evolution is not a directed process in the sense of determinism. It is a stochastic process based on chance events in nature and chance mutations in the organisms. Black circle. Artificial selection by intensive breeding program. Man has created breeds that differ from other breeds, e.g., dogs. Animals are in years. Examples of evolution by anthropogenic action. 1. Antibiotic-resistant microbes. 2. Herbicides-resistant varieties. Allele frequencies in a population are stable and is constant from generation to generation. This is called genetic equilibrium. 3. Pesticide-resistant varieties. 4. The case of industrial melanism. Classical example of natural selection among variants in moth population observed in England. Black circle. The gene pool, total genes and their alleles in a population, remains a constant. Sum total of all the allelic frequencies is 1 and is represented as P plus Q equals 1. P equals frequency of recessive allele, A. Q equals frequency of dominant allele, A. P squared plus Q plus 2PQ equals 1. P equals frequency of homozygous dominant, R. Q equals frequency T parameters before industrialization, 1850s, after industrialization, 1920s, figure homozygous recessive, R. 2PQ equals frequency of heterozygotes, R. When frequency measured differs from expected values, the difference indicates the tree trunk's white covered by lichens became dark due to deposition of soot and smoke. Extent of evolutionary change or disturbance in genetic equilibrium factors affecting Hardy-Weinberg equilibrium. White moths more less 1. Gene migration. When migration of a section of population to another place or population occurs, gene frequencies change in the original as well as in the new population. Melanized moths less more predators feed on melanized moths. White-winged moths when gene migration occurs multiple times. It is called gene flow. Lichens do not grow in polluted area. Pollution indicator, agent of natural selection. Predator, birds emigration, immigration, plus some genes, alleles lost. New genes, alleles added. Black circle moths that were able to camouflage themselves, i.e., hide in the background, survived but no variant is completely wiped out too. Genetic recombination. Variations due to recombination during gametogenesis. 3. Mutation. Microbial experiments show that pre-existing advantageous M. Mutations when selected will result in observation of new phenotypes over few generations. This would result in speciation. 8. A brief account of evolution. Land reptiles were dinosaurs which disappeared. 65. Myoclimatic probable reason form of life appeared around change. 4. Genetic drift. Change in gene frequencies in a small population by chance. Sometimes the change in allele frequency is so different in the new sample of population that they become a different species. The original drifted population becomes founders and effect is called founders effect. 1. Non-cellular forms. Saint 3 BYA evolved in birds. 1. Cellular forms. Saint 2000 mice. Some land reptiles went back to water to evolve fish-like reptiles, e.g. Ichthyosaurus, 200 mile. Invertebrates 500 mile. 5. Natural selection. A process is which heritable variations enabling better survival are enabled to reproduce and leave greater number of progeny. Seaweeds and few plants. 320 mile. Modern-day descendants of reptiles, e.g. turtles, tortoises. Crocodiles possibly evolved right-pointing arrow-lobed finfish, coelacanth, types. Amphibians. Reptiles. Mammals arose from frogs, latex-shelled ancestral extinct reptiles, eggs which do not dry up in sun. Primarily aquatics. Salamanders viviparous, i.e. protect young. Stabilizing selection. Directional selection value other than the mean character value. More individuals acquire aquatic and more individuals acquire mean character value ones inside mothers. Body more intelligent in sensing and avoiding danger. 1938. Fish caught in South Africa was earlier land thought to be extinct. Peak gets higher. A likely reason for restricted distribution of pouched mammals in Australia. Peak shifts in one direction and narrower due to continental drift. When South America joined North America, these animals were overridden by North American fauna. Due to the same continental drift, pouched mammals of Australia survived because of lack of competition from any other mammal. The first mammals were like shrews, whales, dolphins, seals and sea cows are aquatic mammals. Into disruptive selection, more individuals acquire peripheral character value both ends at the distribution curve. Number of individuals. Brief account of evolution of plants. 11. A brief account of evolution. History of vertebrates. Birds, lizards, Tuatara, quaternary turtle, snakes, angiosperms. Lowering plants, monocotyledons, crocodiles, mammals, dicotyledons, quaternary, condors. Cycads, tertiary. 50. Tri-dinosaurs, extinct. Achos, Lycopodium, Cretaceous. 100-150. Jurassic. Therapsids. 200. Hind-type plants, Triassic. Tracheophyte ancestors. Fig. A sketch of the evolution of plant forms through geological periods. The codonts, extinct. 250. Chilopterae ancestors. Permian. Sauropsids. Pelycosaurs, extinct. Synapsids. Carboniferous derivatives. Black circle. Different types of plants evolved in different periods of time. Paleozoic, Mesozoic and Cenozoic. Black circle. Chlorophyte ancestors. Bryophytes. Tracheophyte ancestors. Lycopods. Xylophytans were common ancestors for progymnosperms, ferns, ginkgos, conifers, needles. Sphenopsids. Black circle. Origin of monocotyledons is more recent than that of dioptyledons. Black circle. Giant ferns, pteridophytes, were present, 200 mi, but they all fell to form coal deposits slowly. Fig. Representative evolutionary history of vertebrates through geological periods. Early reptiles, extinct. Derivatives. Turtles, lizards. Snakes and Tuatara arose from common ancestor sauropsids. Decadent ancestors gave way to existing crocodiles and birds and extinct dinosaurs. Story of origin of mammals. Extinct reptiles. Right-pointing arrow. Synapsids. Right-pointing arrow. Pelycosaurs. Right-pointing arrow. Therapsids. Right-pointing arrow. Mammals. Tyrannosaurus. 12. A family tree of dinosaurs. Triceratops. Derivatives. Black circle. Triceratops, three-horned dinosaur with bony frill around back of its head. Pteranodon. Pteranodon were possibly flying reptiles. Archaeopteryx is a transitional fossil between non-avian crocodilian Stegosaurus, large triangular bony plates along the back and spiked tailed dinosaurs and birds. Archaeopteryx. Tyrannosaurus rex, 20 feet in height, had huge fearsome dagger-like teeth. Black circle. Brachiosaurus and Tyrannosaurus arose separately, mostly likely from a common ancestor. Brachiosaurus. Brachiosaurus, long giraffe-like neck, long forelimbs. Stegosaurus. Fig. A family tree of dinosaurs and their living modern-day counterpart organisms like crocodiles and birds. Origin and evolution of man. 13. The skull of baby chimpanzee is more like adult human skull than adult chimpanzees. Among the stories of evolution of individual species, the story of evolution of modern man is most interesting and appears to parallel evolution of human brain and language. Human ancestors years back. Cranial capacity. Specific features. Dryopithecus, Ramapithecus. 15 mi. A more ape-like, more man-like, hairy and walked like gorillas and chimpanzees. Few fossils of man-like bones have been discovered in Ethiopia and Tanzania. Australopithecus. 2 mi. A black circle. 3 to 4 mi. Man-like primates walked in East African grasslands. They were probably not taller than 4 feet but walked upright. Evidence shows they hunted with stone weapons but essentially ate fruit. Homo habilis. 650 cc 800 cc. First human-like being, the hominid, probably did not eat meat. Homo erectus 1.5 Maya 900 cc fossils discovered in Java in 1891. Fig. A comparison of the skulls of adult modern human being. Baby chimpanzee and adult chimpanzee. Neanderthal man 100,000–40,000 years back. 1400 cc. Homo sapiens 75,000 to 10,000 years ago. Ice Age, probably ate meat. Lived in Near East and Central Asia. Black Circle used hides to protect their body and buried their dead. Black Circle arose in Africa and moved across continents and developed into distinct races. Prehistoric cave art 18,000 years ago. One such cave paintings by prehistoric humans can be seen at Bibetka Rock Shelter in Resan district of Madhya Pradesh. Agriculture came around 10,000 years back and human settlements started. Health reflective thought and asserted by Hippocrates along with as per good humor hypothesis arrived at by pure Indian Ayurveda system. Helmanthic health is a state of body and mind where there was a balance of certain humors, e.g. persons with black bile belonged to hot personality and had fevers. Classification of diseases on the basis of transmission: mode of transmission, bacterial, viral, protozoan, air, droplet, aerosol, or object borne, pens, knobs etc. Pneumonia, diphtheria, common cold, smallpox, direct contact, tetanus, smallpox, contaminated food and water, typhoid, dysentery, polio, amoebiasis, insect vector, vector-borne plague, chikungunya, dengue, malaria, body fluids, syphilis, AIDS, trichomoniasis, ascariasis. William Harvey discovered blood circulation experimentally, disproved this good humor hypothesis of health by demonstrating normal body temperature in persons with black bile using thermometer. Filariasis view of biologists in later years. Mind influences our immune system through neural and endocrine systems, and that our immune system maintains our health, i.e., state of complete physical, mental and social and psychological well-being. Health is not simply absence of disease or physical fitness. Vector transmits disease from one organism to another, e.g. female Aedes mosquito is the vector for dengue and chikungunya, while Anopheles spreads malaria. Measures for preventing spread of infectious disease. ES parameters measures personal hygiene, public hygiene, factors affecting health, mental state, genetic, habits, rest and exercise, disorders, infections and lifestyle. Productivity, longevity bring economic prosperity. Infant and maternal mortality are increase. Healthy conditions decrease. Avoid close contact, keeping the body clean. Consumption of clean drinking water, food, vegetables, fruits etc. Proper disposal of waste and excreta. Periodic cleaning and disinfection of water reservoirs, pools, cesspools and tanks. Decontamination of drinking water. Contact with infected persons and belongings should be avoided. Avoid stagnation of water in and around residential areas. Regular cleaning of house. Old coolers. Use of mosquito nets. Introducing larvicidal fishes like gambusia in ponds that feed on mosquito larvae. Spraying of insecticides in ditches, drainage areas and swamps. Right-pointing arrow doors and windows should be provided with wire mesh. Control vectors and their breeding. Disease. It is state of the body when functioning of one or more organ systems is adversely affected, characterized by various signs and symptoms. Types of diseases. Places. Non-infectious, infectious parameters. Transmission from one person to another. Example: AIDS, cancer, AIDS. Pathogens are disease-causing organisms. Most parasites are pathogens living in or on the host multiply and interfere with normal vital activities, resulting in morphological and functional damage. Gut pathogens can survive harsh pH and digestive enzymes. Balanced diet, yoga and regular exercise, personal hygiene. Awareness about diseases and vaccination are very important to maintain good health. Use of vaccines and immunization programs have enabled us to completely eradicate a deadly disease like smallpox. Large number of infectious diseases like polio, diphtheria, pneumonia and tetanus have been controlled to a large extent by the use of vaccines. Biotechnology is at the verge of making available newer and safer vaccines. Discovery of antibiotics and various drugs have enabled us to effectively treat infections. AIDS, acquired immunodeficiency syndrome 1 reported 1981. Killed approximately 25 million people in last 25 years. It is deficiency of immune system, acquired during the lifetime of an individual. Syndrome means, group of symptoms non-congenital. Fatal infectious disease causative agent HIV, human immunodeficiency virus enveloped virus enclosing RNA genome high-risk individuals mode of transmission sexual contact multiple sexual partners mother to fetus blood transfusion repeated blood transfusion drug addicts intravenous infected needles macrophages helper T cells entry into body cells viral protein coat with RNA plasma membrane animal cell 8 cancer dreaded non-infectious disease. Major cause of death all across the globe. Parameters normal cells cancerous cells, neoplastic cells cell growth and differentiation highly controlled and regulated. Uncontrolled and non-regulated contact inhibition present, virtue of which contact with other cells inhibits their growth lost. So these cells keep on dividing and form mass of cells called tumor. Neoplasm life cycle entry of virus in body plus placenta viral RNA is introduced into cell cytoplasm viral DNA is produced by reverse transcriptase hence named retrovirus viral DNA incorporates into host genome new viral RNA is produced by the infected cell types of tumor parameters benign malignant tumor cancer location confined to original place grow rapidly and spread to other parts damage little damage invade and damage other cells starving normal cells by competing for vital nutrients metastasis no yes. Cells sloughed from such tumors reach distant sites through blood and start new tumor called metastasis, most feared property, cause, normal cell cancerous cell neoplastic transformation proto-cellular. Oncogene, CUNC, oncogenic causative agents called carcinogens ionizing radiations X-rays. Y-rays non-ionizing radiations UV rays. Physical agents, chemical agents in tobacco smoke, biological agents oncogenic viruses, carry viral oncogenes, diagnosis, detection. Technique early detection allows the disease to be treated successfully in many cases. Detect basis biopsy histopathological studies changes in tissue blood and bone marrow test cell counts leukemias radiography X-rays internal organ cancers computed tomography CT X-rays internal organ cancers 3D image magnetic resonance imaging MRI strong magnetic fields and non-ionizing radiations accurately detect pathological and physiological changes in living tissue molecular techniques identification. of genes responsible for susceptibility to certain cancers, antibodies based against cancer-specific antigens. Certain cancers tumor cells have ability to avoid detection and destruction by immune system. Approaches for treatment: surgery, immunotherapy, interferons, biological radiotherapy. Tumor cells are radiated lethally. Response modifiers activate immune system and helps in destroying the tumor. Chemotherapy. Side effects like hair loss, anemia. New viruses are produced. Nucleus DNA. New viruses can infect other cells. Fig. Replication of retrovirus sequence of events. Black circle infected cells, macrophages, can survive while viruses are being replicated and released, hence called HIV. Factory HIV enters into macrophages and T helper cells, T. Simultaneously black circle, there is progressive decrease in number of helper T cells. Initial symptoms: bouts of fever, diarrhea, weight loss. Later the immunodeficient patient is prone to infections, especially mycobacterium, viruses, fungi, toxoplasma etc. There is always a time lag between infection and appearance of AIDS symptoms. This may vary from a few months to many years, usually 5 to 10 years. Treatment: antiretroviral drugs can only prolong life but cannot prevent death. Diagnostic test: ELISA, enzyme-linked immunosorbent assay. Prevention: different agencies like NGOs, NARCO. WHO started number of programs to educate, make people aware of AIDS, don't die of ignorance, and some of the measures preventing spreading of HIV infection, making blood banks safe from HIV, greater than use of only disposable needles and syringes in public and private hospitals and clinics, free distribution of condoms. Advocating safe sex, controlling drug abuse. Immunity: the ability of the host to fight the disease-causing organisms conferred by the immune system is called immunity. Types: parameters: innate, observed from time of birth, exposure to infection not required, defense non-specific, memory record acquired after birth, required specific to innate immunity. This immunity is accomplished by providing different types of barriers to the entry of the foreign agents. Types of barrier: physical structures involved. Barrier: basic function, skin, prevent entry of microbes, mucus coating of the epithelium lining the respiratory, gastrointestinal and urogenital tracts. Right pointing arrow: trap microbes entering our body. Physiological: saliva in the mouth, acid in the stomach, tears from eyes, prevent microbial growth. Cellular neutrophils, PMNL monocytes. Macrophages: natural killer cells. Type of lymphocytes: phagocytose. Microbes destroy microbes. Cytokine: interferons produced by virus-infected cells that protect non-infected cells from further infection. Memory-based immunity evolved in higher vertebrates. Notes: lymph immune, RESPONSEACQUIRED. Immunity: the human immune system consists of lymphoid organs, tissues, cells and soluble molecules like antibodies. This response is carried out by two special types of lymphocytes present in our blood: band T lymphocytes, primary, first time type; secondary, anamnestic, subsequent times exposure to origin and or maturation of lymphocytes. Primary lymphoid organs: thymus, bone marrow. Pathogens: intensity: lymphocytes become antigen sensitive. Low high matured lymphocytes. T cells migrate. B cells, based on memory of first encounter, provide microenvironments for development and maturation of lymphocytes. These responses are carried out by B and T lymphocytes. Secondary lymphoid organs: spleen, lymph nodes, tonsils, appendix, M. Alt. Pairs patches provide sites for interaction of lymphocytes with antigen which proliferate to become effector cells. Antigen binding site. Each antibody has 4 peptide chains, HL2. Long heavy chains help T cells mediated response. B cells produce antibodies. Army of proteins in blood. Thymus. Black circle. 2 short light chains called immunoglobulins, light chain types. IgA, IgM, IgE, heavy chain lymphatic vessels. Cell mediated immune. Humoral immune response, antibody response. CMI mediated immune response. Fig. Structure of an antibody. Moleculate lymphocytes are responsible for graft rejection. Tissue and blood group matching are essential before undertaking any graft, transplant, and even after this patient has to take immunosuppressants throughout life, if the pathogens succeed in gaining entry to our body. Specific antibodies and T cells serve to kill these pathogens. Vaccination and immunization. Types of immunity structure. Bone marrow. Thymus. Antibodies active, produced within the host body. Passive. Ready-made, preformed antibodies are directly given. Longer shorter spleen time taken for full, effective response. Lymphoid structures. Organs. Typical main lymphoid organ where all blood cells are produced, including lymphocytes. Lobed organ located near the heart and beneath the breastbone. Quite large at the time of birth, keeps reducing in size with age, and by the time puberty is attained it is reduced to a very small size. Large bean-shaped organ. Mainly contains lymphocytes and phagocytes. Acts as a filter of the blood by trapping blood-borne microorganisms. Large reservoir of erythrocytes. Lymph nodes. Small solid structures located at different points along the lymphatic system serve to trap the microbes, antigens which happen to get into the lymph and tissue fluid. Antigens trapped in the lymph nodes are responsible for the activation of lymphocytes present there and cause the immune response. Marked right pointing arrow. Mucosa-associated lymphoid tissue is located within the lining of major tracts like respiratory, digestive and urinogenital tracts. Constitutes about 50% of lymphoid tissue in human body. Memory cells. Examples: fetus. Natural antibody production. Infection in host. Vaccination. Deliberate injection of living dead microbes. Proteins. Placenta. Mother. Colostrum. Mother. IgA. Infant immunization. Principal property of memory of immune system. Active preparation of antigenic proteins of pathogen. Inactivated, weakened pathogen introduced into body. Passive direct preformed antibodies. Antitoxin is injected, such as antitoxin against snake bites. Allergies. Exaggerated response of immune system to certain antigens present in the environment. Allergen substances to which exaggerated immune response produced, e.g. pollens, mites in dust, animal dander, etc. is antibodies. IgE type symptoms: sneezing, watery eyes, running nose, difficulty in breathing. Chemical, histamine and serotonin from mast cells released. Diagnosis: patient is exposed to or injected with very small doses of possible allergens, and reaction studied. Treatment: antihistamine. Adrenaline and steroids quickly reduce the symptoms of allergy. The antibodies produced in the host body against antigens would neutralize the pathogenic agents during actual infection. The vaccines also generate memory B cells and memory T cells that recognize the pathogen quickly on subsequent exposure and overwhelm the invaders with a massive production of antibodies. Recombinant DNA technology. has allowed the large-scale production of antigenic polypeptides of pathogen in bacteria, yeast. Hence greater availability for immunization, e.g. hepatitis B vaccine produced from yeast. Effects of modern-day lifestyle. Protected environment provided early in life has resulted in lowering of immunity, and person is more sensitive to allergens more. And more children in metro cities of India suffer from allergies and asthma due to more sensitivity to the environment. Autoimmunity. Memory-based acquired immunity evolved in higher vertebrates can distinguish foreign molecules as well as foreign organisms, pathogens, from self-cells. If lost this ability, results self-destruction, body attack self-cells. Reason genetic, unknown. Example rheumatoid arthritis. Drugs and sportsperson. 9 abuse chemical when taken for a purpose other than medicinal use or in amounts, frequency impairs one's physiological or why to use, psychological functions and constitutes drug abuse. Source majorly from flowering plants and some from fungi. Increase muscle strength and bulk greater than. Promote aggressiveness. Commonly abused drugs are enhance athletic performance. Drug receptors. Source intake. Examples action and anything specific. Commonly abused drugs narcotic, analgesics, opioids. CNS GIT latex of poppy plant, Papaver somniferum S. Knotting. Injection morphine. Effective sedative and painkiller useful in patients undergone surgery. Greater than diuretics. Heroin, smack, diacetylmorphine, depressant and slows down body functions. Anabolic steroids. Odorless, white, bitter crystalline compounds. Certain hormones. Common side effects. Increase aggressiveness. Opium poppy. Chemical structure of morphine. Mood swings. Cannabinoids principally in brain. Inflorescence, flower tops, leaves and resin of cannabis plant. Cannabis sativa. Inhalation, oral churus. Ingestion. Effects on cardiovascular system of the body. These days cannabinoids are also being abused by some sportspersons. Right pointing arrow. Depression, hashish, ganja, marijuana. Stunted growth because of premature closure of growth centers of long bones. Severe facial and body acne. Typical side effects. Homale leaves of cannabis sativa. Skeletal structure of cannabinoid molecules. Stimulants. Science. Breast enlargement. Decreased sperm production. Reduction in size of testicles. Acne, premature baldness, enlargement. Prostate gland. Potential for liver and kidney dysfunction. Female coca plant. Erythroxylum coca, native of South America. Snorting, cocaine, coca alkaloid. Right pointing arrow. Commonly called coke, crack, interferes with transport of neurotransmitter dopamine. Potent stimulating action on CNS, producing sense of euphoria and increased energy. Excessive dosage causes hallucinations. Masculinization, features like males, hallucinogens. Atropa belladonna, datura. Abnormal menstrual cycles. Enlargement of clitoris. Excessive hair growth on face and body. Deepening of voice. Other drugs have been used for hundreds of years in folk medicine. Religious ceremonies and rituals all over the globe help patients cope with mental illness like depression, insomnia, synthetic barbiturates, benzodiazepines, amphetamines, flowering branch of datura. These side effects may be permanent with prolonged use. Paves the way to hard drugs. Smoked jute. Snuff. Chemical substance. Action of nicotine. An alkaloid stimulates adrenal gland to release adrenaline and non-adrenaline into blood circulation. Effects: respiratory system. Increases carbon monoxide (CO) in blood and reduces concentration of hembound oxygen. causes oxygen deficiency in the body, circulatory system, increase heart rate and blood pressure. Common diseases are bronchitis, emphysema, coronary heart disease, gastric ulcer, risk of cancers, oral cavity, 5 throat, unexplained absence from school, college, 5 lungs, urinary bladder. Tobacco has been used by humans for more than 400 years. Packets of cigarettes warns against smoking and says how it is injurious to health. 12 addiction and dependence. Addiction because of perceived benefits. Drugs are frequently used repeatedly that leads to psychological attachment to certain effects like euphoria and temporary feeling of well-being. Dependence. It is the tendency of the body to manifest a characteristic and unpleasant withdrawal syndrome. If regular dose of drugs, alcohol is abruptly discontinues, addiction drive people to take drug even when its use becomes self-destructive with repeated use of drug. Tolerance level of receptors increases. Receptors respond only to higher doses of drugs, leading to greater intake. Effects of drug, alcohol abuse, reckless behavior, vandalism, violence, depression, fatigue, drop in academic performance. Warning signs: poor personal hygiene, withdrawal, isolation, aggressive and rebellious behavior, loss of interest in hobbies, change in sleeping and eating habits, fluctuations in weight and appetite, deteriorating relationships with family and friends. High doses lead to coma and death due to respiratory failure, heart failure or cerebral hemorrhage. Right pointing arrow: chronic use of drugs, alcohol damage nervous system and liver, cirrhosis. Use of drugs during pregnancy adversely affect fetus, some far-reaching implications. Abuser may turn to stealing. Addict becomes the cause of mental and financial distress to entire family and friends. Withdrawal syndrome: if drug is abruptly discontinued. Symptoms include anxiety, nausea, shakiness, sweating. In severe cases, can be life-threatening. Person needs a medical supervision. Prevention and control: prevention is better than cure. Avoid undue peer pressure on child related to studies, sports or other activities. Education and counseling: channelize energy of child into healthy pursuits like sports, yoga, reading, music, etc. Sort out problems by seeking help from parents and peers. Looking for danger signs: alert parents. Teachers and close friends need to look for and identify the danger signs of substance, drug, alcohol abuse, and appropriate measures would then be required to diagnose the malady and underlying cause. Proper remedial steps or treatment should be taken by seeking professional and medical help in the form of highly qualified psychologists, psychiatrists and their addiction and rehabilitation programs. This will totally relieve the individual from these evils. Adolescence and abuse: adolescence means both a period and a process during which a child matures in terms of his/her attitudes and beliefs for effective participation in society. Adolescence is a bridge linking childhood and adulthood. It's a period between 12 to 18 years of age. A vulnerable phase of mental and psychological development of an individual. It is accompanied by several biological and behavioral changes: curiosity, need for adventure and excitement, and experimentation. Motivate youngsters towards drug and alcohol use. First use may be out of curiosity but later used to escape from stress. Pressures to excel in academics. Perception that it is cool. Television, movies, newspapers, internet promote this perception. Unstable or unsupportive family structures and peer pressure also promote drug and alcohol abuse. user into a vicious circle leading to their regular use, abuse. Use of drugs even once can be forerunner to addiction, and pull the animal husbandry stringent cleanliness hand and management of farm and farm animals a professional approach to boost our food production. Some of the management procedures employed in animal black circle practices involve effective housing, dairy farm, poultry farm, adequate water farm system or management. Cattle maintain disease-free one. Dairy farm management, dairying, management of animals for milk and its products for human consumption, e.g., cows. Buffaloes conditions cattle diet, fodder, Jersey quality up pointing arrow, oil cake rich, quantity balanced aimed at increased mechanization in dairy farming, particular milking, storage and transport of milk. Reduces chance of direct contact of the produce with the handler, increasing yield depends on quality of breed, improving quality of milk. Definition: black circle agricultural practice of breeding and raising livestock useful to humans. Some of the products and the animals involved are useful products: source milk, cows, buffaloes, goats, eggs, poultry, birds, chicken, ducks, turkey, geese, meat, cattle, sheep, pigs, wool, sheep, silk, silkworms, honey bees, regular inspection, bee, veterinary doctor, high yielding potential under given climatic conditions, resistance to diseases. To poultry farm management involves use of birds for food, eggs, meat. More than 70% population of livestock is in India and China, but contribution to world's farm produce is 25%, that result in problem productivity per unit is low, insufficient food supply due to ever-increasing population size. Solution: improvement in conventional practices of animal breeding by applying biological principles. Application of newer technologies to animal husbandry. Extension of poultry farming, birds, fowl, chicken, ducks, turkey, geese practices involved Leghorn selection of disease-free and suitable breeds, black circle, proper and safe farm conditions, proper feed and water, maintaining proper health care and hygiene, beekeeping, apiculture. It is the maintenance of hives of honey bees for the production honey, age-old, high-income yield cottage industry and is not labor-intensive. Most common bee in India, Apis indica products obtained from honey bee. Advantages: A. Increases product yield. B. Enhances crop yield. Bees wax honey. Salient points for preparation food. High successful beekeeping of cosmetics. Nutritive value. I. Knowledge of the nature and habits of bees and polishes used in indigenous medicine. E. Selection of suitable location for keeping the beehives. Bees are effective pollinators. E. Catching and hiving of swarms, group of bees, areas for practice. IV. Management of beehives, pastures for shrubs, crop orchards, fields during different seasons. V. Handling and collection of honey and beeswax. Brassica, apple, pear. Animal husbandry, fisheries. Disease, bird flu cause symptoms. H. N. Virus, respiratory difficulty, fever. Malaise, fruit fisheries, rearing fish to catching, processing and selling mollusks, shellfish, crustaceans, prawns, crabs. Aquatic animals industry devoted of aquaculture, pisciculture, enhancement in aquatic yield including plants and animals, increase in total fish yield. Products from fishery food, lobster, prawn, fish. Edible oyster, people at risk, poultry farmers exposed to infected birds. People who eats undercooked eggs, poultry, sunflower, fresh water, FW, Katla, Rohu, common carp, marine, M, Hilsa, sardines, mackerel. Pomfrets to meet the increasing demand on fisheries. Different techniques have been employed that led to Blue Revolution. Keeping beehives in crop fields during flower period increases pollination efficiency and yield. Animal breeding breeding or hybridization. Types of selective breeding. Breed a group of animals related by descent and similar in most characters like general appearance, features, size, configuration etc. Aims of animal breeding: increase the product yield, quantity 1, black circle improving desirable qualities in produce and cattle high yielding variety 1. Outcrossing mating of animals within the same breed but having no common ancestors up to 4 to 6 generations down. Pointing arrow advantage: a single outcross helps increase productivity of animals below average up pointing arrow growth rate. In beef cattle up pointing arrow milk production, right pointing arrow disease resistant high reproductive rate longer productivity span 3. Interspecific hybridization controlled breeding experiments. Male and female of two different related species are mated. Artificial insemination AI identify elite bull multiple ovulation embryo. Transfer MOE identify superior cow. Inbreeding outbreeding mating of more closely related individuals within the same breed for 4 to 6 generations. Mating between male and female of same breed. D. No recent common ancestor or different breeds or different species. Inbreeding increases homozygosity and is useful to evolve pure lines too. Crossbreeding right pointing arrow mating between superior male of one breed and superior female of another breed. Advantage: increases the productivity of population as superior genes are accumulated and harmful recessive genes are eliminated. This method allows combination of superior qualities of two different breeds on a commercial scale. Down pointing arrow advantage: stable new breeds superior to existing breeds can be developed. Disadvantages greater than inbreeding. Example: a new breed of sheep, male, female Merino Ram X Bikaneri U depression, ID. Due to continued close inbreeding, fertility productivity reduces. Hisardale location. Punjab inject with FSH-like hormone advantage. Progeny may combine desirable features of both the parents and may be of considerable economic value. Superovulation, 6 to 8 over cycle, extract semen either used immediately or cryopreservation, cold storage, or natural insemination. Example: non-surgical extraction of embryo. Gestation in surrogate cow inject into reproductive tract of superior cow, male. Donkey plus female horse, mare. Advantages: mule, sterile, greater chances of fertilization. Economically relevant as all sperms can be used since semen from one bull can fertilize more than one cow. If needed, helps to overcome several problems of normal mating, increase herd size and quality in less time. Genetic mother available for another round of superovulation. Inbreeding depression can be overcome by outcrossing, i.e. method to restore fertility and yield. Disadvantages: MOA is demonstrated on mammals such as cattle, mares, sheep, rabbits. Right pointing arrow superior cow, high yield of milk, lactation. Superior bull. Lean meat with less lipid have been bred successfully to increase herd size in a short time. The success rate of crossing not applicable to fishes and birds mature and is fairly low. Introduction: principles of biotechnology. Core techniques involved in modern biotechnology. Biotechnology deals with techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. Parameters definition genetic engineering bioprocess engineering techniques to alter the chemistry of genetic material to introduce these into host organisms and thus change the phenotype of host organism maintenance of sterile ambience chemical engineering processes to enable growth of only the desired microbe, eukaryotic cell in large quantities parameters traditional biotechnology modern biotechnology organisms involved microbes genetically modified organisms large-scale production small-scale include manufacture of biotechnological products like antibiotics, vaccines enzymes, etc. Creation of rDNA gene cloning gene transfer examples, technique include curd, bread or wine in vitro fertilization, leading to a test tube baby making the ability to multiply copies of antibiotic resistance gene in E. coli was called cloning of antibioresistance gene in E. coli. EFB, European Federation of Biotechnology, synthesizing a gene and using it the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. Developing a DNA vaccine 3 basic steps in genetically modifying organisms. It encompasses both traditional view and modern molecular biotechnology, correcting a defective 1. Identification of DNA with desirable genes, 2. Introduction of the identified DNA into the host gene, 3. Maintenance of introduced DNA in the host and transfer of the DNA to its progeny advantages of biotechnology over other TECHNIQUESKEY tools of recombinant DNA technology methods advantage disadvantage 1. Enzymes 2. Vectors 3. Competent host cells nucleases 1. Asexual reproduction preserves genetic information no variations enzymes most commonly used enzymes in genetic engineering are DNA polymerase ligases E. Sexual reproduction provides opportunities for variations and formulation of unique combinations of genetic setup some of which may be harmful to the organism as well as the population nucleases. Catalyze the cleavage of nucleic acids types exonucleases endonucleases palindromic sequence reads same on the two strands, from 5 to 3 and 3 feet 5 feet direction, when orientation of reading is kept same e. Traditional hybridization used in plant and animal breeding very often lead to inclusion and multiplication of undesirable genes along with desirable genes. Remove nucleotides from the ends of DNA make cuts at specific positions within the DNA i.e. at recognition, palindromic sequence in the year 1963. The two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated IV. Genetic engineering allows us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into target organism. Methylase add methyl groups to bacterial DNA restriction endonuclease. Molecular scissors cut the DNA of bacteriophage enzymes restriction endonuclease VECO religase genus Escherichia coli species more than 900 restriction enzymes ha. We've been isolated from over 230 strains of bacteria, prokaryotic cell, each of which recognize different recognition sequences, strain order of Re13 isolation when source DNA and vector DNA are cut by the same restriction enzyme. The resultant DNA fragments have the same kind of sticky ends. Sticky ends are named so because they form hydrogen bonds with their complementary cut counterparts, and this stickiness facilitates the action of the enzyme DNA ligase. Nomenclature. Naming of enzyme Weacher DNA. Functions by. For DNA stay in sticky and. Inspecting the length of DNA sequence. Binds to the specific recognition sequence. Cuts the two strands of dsDNA at specific points in their sugar phosphate backbones and leaves single-stranded portions at the ends. These overhanging stretches and called sticky ends. Ligase first. Restriction endonuclease Hind II. Isolated and characterized 5 years later. Recognizes sequence of 6BP first recombinant DNA was prepared by Stanley Cohen and Herbert Boyer. 1972. Antibiotic-resistant gene introduced recombinant plasmid Escherichia coli plasmid of Salmonella typhimurium into steps in formation of rDNA 7 cloning vectors. Vectors are vehicles for delivering foreign DNA into recipient cells. Vectors used at present are engineered in such a way that they help easy linking of foreign DNA and selection of recombinants from non-recombinants features of cloning. Vectors SAD phase 1. Origin of replication, ORI, sequence from where replication starts. 2. Responsible for controlling copy number of the linked DNA. Right pointing arrow. Those vectors are preferred which support high copy number. 2. Selectable marker, transformation. Procedure through which piece of foreign DNA is introduced in a host bacterium. Black circle insertional inactivation. Insertion of GOI within antibiotic resistance gene. Selectable marker results in inactivation, formation of the coded product. Hypothesis. Insertion of GOI at BAM high site in TET. If transformation fails, non-transformants are obtained in antibiotic-lacking agar medium but they don't grow on antibiotic-rich medium. If transformation successful, transformants obtained are of two types. Stamp PBr322 Ampsal. All transformants are not recombinants but all recombinants are transformants. Non-recombinants recombinants insertional inactivation ECORCA Hind ligate 1 antibiotic resistant gene helps in selecting the transformants E. coli cloning, whereas the other antibiotic vector PBr322 foreign DNA at BAM high site resistant gene helps in selection of recombinants ROP codes for the proteins involved in the replication of the plasmid gene of interest. Cloned resistance to ampicillin, resistance to tetracycline, due to inactivation plasmids as vectors. Extrachromosomal, circular, double-stranded DNA, black circle replicate independent of the control of chromosomal DNA, autonomously. They may have 1 or 2 copies per cell or even 15 to 100 copies per cell. Helps in selection of transformants. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloramphenicol, tetracycline or kanamycin, etc. are considered useful selectable markers for E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. 3. Cloning sites. Restriction sites. Single recognition site for a restriction enzyme within the vector is a preferable feature. Greater than presence of more than one recognition sites within the vector will generate several fragments, which will complicate the gene cloning. The ligation of alien DNA, gene of interest, GOI, is carried out at a restriction site present in one of the antibiotic-resistant genes. Isolation of the genetic material, DNA. 10 methods of transformation in majority of organisms. DNA is the genetic mater. Since DNA is enclosed within the membranes, have to break the cell open to release DNA along W other macromolecules. Bacteria, right pointing arrow, lysozyme. Fungi, right pointing arrow, chitinase. Plant cell, right pointing arrow, cellulase. In order to get DNA 1 microinjection, black circle recombinant DNA is directly injected into the nucleus of an animal cell. Bialistic, gene gun, other cloning vector, selection of recombinants due to inactivation of antibiotic-resistant gene as in PBR 322 is a cumbersome procedure because it requires simultaneous plating of two plates having different antibiotics to overcome the disadvantage of PBR 322. Alternative selectable markers, LAC-Z, acting as reporter enzyme, have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of chromogenic substrate. LACZ gene coding for B-galactosidase acts as selectable marker in the plasmid experiment. Insert foreign DNA at LAC-Z gene plus transformation in E. coli. Chromogenic substrate. Plant cells are bombarded with high-velocity microparticles of gold or tungsten coated with DNA. 3. Heat shock method. Pure form, free from other macromolecules. It treated with different enzymes like NAS, protease etc. IV. Disarmed pathogen. Vector chilled ethanol. Pure right pointing arrow centrifuge to precipitate DNA. DNA spooling 11 competent host for transformation with recombinant DNA. DNA is hydrophilic, so it cannot pass through cell membranes. In order to force cell to take up alien, it must first be made competent by treating with ice-cold calcium chloride. Entry of rDNA in host cell is due to transient pause created by heat shock, 42 degrees Celsius, and not due to Ca, ions. Fails succeeds. Divalent cations increases the efficiency with which DNA enters the bacterium through pores in its cell wall. 2. Fragmentation by restriction. Endonucleases. Separation and isolation of DNA fragments. Gel electrophoresis. Black circle separation of negatively charged DNA molecules under an electric field through a medium, matrix. Black circle most commonly used matrix for DNA separation is blue-colored colonies, white-colored colonies. Non-recombinants. Recombinants 12 process of recombinant DNA technology. Isolation of DNA natural polymer obtained from seaweeds. Agarose separate DNA fragments through sieving. Effect fragmentation of DNA by restriction. Endonucleases wells filled with DNA fragments. Separation on the basis of size, smaller the DNA fragment farther. DNA bands smallest electrodenote. Isolation of desired DNA fragment. Electrophoresis. Cathode 0000 amplification of gene of interest. PCR. T plasmid of Agrobacterium tumefaciens. Agrobacterium tumefaciens. A pathogen of several dicot plants is able to deliver a piece of DNA known as tDNA to transform normal plant cells into a tumor and direct the tumor cells to produce the chemicals required by the pathogen. Black circle disarmed tumor-inducing T plasmid is used, which is no more pathogenic to the plants but is still able to use the mechanism to deliver the genes of our interest into varieties of plants. Bacteriophages. High copy number than plasmid. Retroviruses. Black circle retroviruses in animals have the ability to transform normal cells into cancerous cells. Black circle disarmed retroviruses are used to deliver desirable genes into Animal cells stained with gel ligation of the DNA fragment into a vector exposed ethidium bromide UV rays process elution removal of DNA bright orange fragment from gel bands transferring the alien DNA, recombinant DNA into the host culturing the host cells in a medium at large scale, bioreactors, extraction and purification of the desired product. Purified DNA fragments are generally amplified, PCR. Before constructing rDNA by joining with cloning vector. NPCR polymerase chain reaction in vitro amplification of DNA, gene of interest, reaction mixture work, function formation of DNA chain nucleotides to sets of chemically synthesized oligonucleotides. Complementary to the regions of DNA primers Taq polymerase thermostable DNA polymerase, isolated from bacterium, Thermus aquaticus, remains active during high temperature induced denaturation of dsDNA. It extends the primers i.e. meant for chain elongation, template DNA for gene of interest via culturing of recombinant host cells, biosynthetic stage. The cells harboring cloned genes of interest may be grown in bioreactors. Vessels in which raw materials are biologically converted into specific products using microbial plant, animal human cells and provide optimal growth conditions, temperature, pH, substrate, salts, vitamins. Oxygen, laboratory, bioreactors parameters laboratory bioreactors culture small volume large volumes, 100-1000 hits, maintaining optimal conditions not possible growth rate of cell never. Optimum production small scale large scale cylindrical or with curved base facilitate mixing of reactor contents stirrer facilitate even mixing and oxygen availability throughout the bioreactor. Commonly used bioreactors are stirred type having agitator system, oxygen delivery system, pH control system, foam control system, sampling ports to withdraw small volumes of culture periodically. Types of stirred tanks: genome DNA sequence of events region to be amplified 31 deciseconds DNA 5 steps denaturation heat 3 simple stirred tanks parched stirred tank primers annealing 5 feet increased surface area for oxygen transfer in open culture system. Continuous culture system used medium is drained out from one side while fresh medium is added from the other. To maintain the cells in their physiologically most active log, exponential phase, larger biomass higher yields of desired protein, motor DNA polymerase, Taq polymerase, plus deoxynucleotides gas. Entrainment ACID, base for pH control foam breaker flat-bladed impeller culture broth extension stream for sterilization 30 cycles, process repeated, n times bubbles, dramatically increase the oxygen transfer. Area sterile air amplified, -1 billion times. The amplified fragment if desired can now be used to ligate with a vector for further cloning. Ligation of the DNA fragment into a vector by DNA ligase via downstream processing right pointing arrow separation and purification of the desired product, recombinant protein from heterologous host, non-native host, product has to be formulated with suitable preservatives. Strict quality control testing is done for each product. The downstream processing and quality control testing vary from product to product. IX product is subjected for marketing as a finished product via insertion of recombinant DNA into the host cell. Transformed host cells are selected with the help of selectable marker genes, Biotechnological applications in medic advantages of recombinant therapeutics, Black Circle 30. Recombinant the rape have been approved for WHO use the world over. In India of these are presently being marketed. Mass production of safe and effective drugs. Do not induce unwanted immunological responses. Genetically engineered Hun insulin. Problem. Insulin extracted slaughtered cattle and pigs cause allergy. Solution. Production of HUM1 introductionist resistant plants. Choice of gene depends on biotechnology. Essentially deals with industrial-scale production of biopharmaceuticals and biologicals using GM microbes, fungi, plants and animals. Provides resistance to insects without the need. I. Target pest. E. Crop. Black circle Krill and Krill bright pointing arrow cotton bollworm for insecticides. Biopesticide. Examples of biopesticides are Bt. Cotton. Bt. Black circle Krull ab right pointing arrow com borer corn, rice, tomato, potato and soybean etc. Bt toxins are insect group specific. Applications of biotechnology include Bt cotton Lepidoptera, tobacco bootworm, therapeutics diagnostics processed food Bacillus thuringiensis, a bacterium, source of the cry-gene army worm. Cotton bollworm bioremediation genetically modified waste treatment Coleoptera, beetles black circle Diptera. Flies and mosquitoes crops for agriculture energy production codes for Bt toxin produced during a inactive protoxins, protein particular phase of crystals, their growth or pest resistant plants 3. Critical research areas of biotechnology. Method of cellular defense seen in all eukaryotes against pest infestation. Endotoxin sequence of events. Providing best catalyst in the form of improved microbes or pure enzymes technique responsible. RNA interference, NA, cry gene from Bacillus thuringiensis, creating optimal conditions through genetic engineering. Bt cotton cells express inactive protoxins, black circle based on post-transcriptional silencing of mRNA. Downstream processing technologies for purification ingested by cotton bollworms. Black circle translation of mRNA coded from pest-specific genes is silenced, prevented due to formation of complementary dsRNA source alkaline pH in the midgut of insects. Solubilisis protein crystals 2BIOTECHNOLOGICELAPPLICATIONS in agriculture. Food production could possibly be increased. Active toxin viruses with mobile genetic elements transposons replicating create pores in midgut epithelial cells. RNA genome be 3 ways via an RNA intermediate. The case of nematode resistant transgenic tobacco sad phase 1. Agrochemical based agriculture swelling of midgut epithelial cells and thereby lysis. E. Organic agriculture death of insect black circle pest causing roots knot disease in tobacco plant. Meloidogyne incognita, nematode, helminth. I. Genetically engineered crop-based agriculture. Genetically modified organisms. Green revolution resulted in tripling of food production, greater than organisms including plants, bacteria, fungi and animals whose genes have been altered by manipulation are called genetically modified organisms. GMO. Nematode-specific gene is introduced in host plant, tobacco, by using T. plasmid, vector, of Agrobacterium tumefaciens, in such a manner that it produces both sense and antisense RNA in the host cells. Reasons for success of Green Revolution. Improved crop varieties. Applications of genetic modification. 1. made crops more tolerant to abiotic stresses, cold, drought, salt, heat, agrochemicals, fertilizers plus pesticide, better management practices, reduced reliance on chemical pesticide, pest-resistant crops, sense RNA and antisense RNA being complementary form. dsRNA that initiates RNAi, problem area and hindrances SAD phase, help to reduce post-harvest losses. IV, increased efficiency of mineral usage by plants, prevents early exhaustion of black circle. Enhancement in food production by Green Revolution was still not enough to feed growing population. Fertility of soil. Enhanced nutritional value of food, e.g. golden rice, i.e. vitamin A, enriched rice. Parasite could not survive in a transgenic host expressing specific interfering RNA. Agrochemicals are often too expensive for farmers of developing world. Tailor-made plants. Plants have been developed to supply alternative resources to industries in the form of starches, fuels and pharmaceuticals. Increase in yield with existing varieties is not possible using conventional breeding host. Peptide maturation of proinsulin to insulin. Recombinant insulin manufacture by Eli Lilly, an American comp in 1983. The main challenge for product of insulin using rDNA technique was getting insulin assembled into a mature form. Molecular diagnosis method. Sequence of events, artificially synthesized DNA sequence for a chain, DNA sequence for B chain parameters, early detection greater than examples, conventional not possible, serum and urine analysis, modern possible, RDT, PCR, ELISA, plasmid E. coli chain A, coli PCR, polymerase chain react. Iron, bases, nucleic acid amplification, black circle advantage, detection of very low concentration of a bacteria or virus, uses, detection of HIV infection, mutations in genes in cancer patients. Genetic disorders. Chain B produced separately, extracted and combined by creating disulfide bonds. ELISA, enzyme-linked immunosorbent assay. Bases, antigen-antibody interaction. Black circle uses, detect the presence of antigens or antibodies synthesized against pathogens. Human insulin autoradiography probe, radioactive ssDNA or ssRNA that hybridizes with complementary DNA. Probe will not hybridize with mutated gene. Hence mutated gene will not appear on the photographic film due to lack of complementarity. Insulin is a peptide hormone and can be degraded by proteases in our gut. Gene. The rapid insertion of genes into an individual's cells to treat diseases by replacing a defective mutant allele with a functional one. Gene targeting, which involves gene amplification. First clinical gene therapy was conducted in 1990. In a 4-year-old girl to treat adenosine deaminase, ADA, deficiency. ADA enzyme is crucial for immune system to function. Transgen IC animals possess manipulated DNA and express foreign gene. Transgenic rats, rabbits, pigs, sheep, cows and fish have been produced. 95% of transgenic animals are mice. Uses of transgenic animals to study how genes are regulated and how they affect the normal functions of body, e.g. study of insulin-like growth factors. Transgenic models exist for study of diseases like cancer, cystic fibrosis, rheumatoid arthritis and Alzheimer's. Treatment for ADA deficiency. 1. Enzyme replacement therapy. Functional ADA is given by injection, not completely curative. 2. Bone marrow transplantation in children. 3. Gene therapy could be a permanent cure if bone marrow transplantation is done at early embryonic stages. Steps in gene therapy: lymphocytes of patient biological products, -1 antitrypsin, to treat emphysema, black circle. Similar attempts are made for treatment of PKU, phenylketonuria, and cystic fibrosis, black circle. First transgenic cow Rosie developed in 1997, producing human protein-enriched milk, 2.4 grams per liter. The milk contained alpha-lactalbumin, more balanced product for human babies than natural cow milk. Black circle vaccine safety: transgenic mice are being used to test the safety of polio vaccine to replace the use of monkeys. Chemical safety testing: black circle transgenic animals are made more sensitive to toxic substances to obtain results in less time. ADA cDNA retrovirus-mediated transformation: genetically engineered lymphocytes, not immortal, infused in patient. Ethical issues: black circle genetic modification of organisms, GMO, can have unpredictable results when such organisms are introduced into the ecosystem. Genetic manipulation of living organisms by humans has to regulated for moral and biological significance. Greater than GEAC, Genetic Engineering Approval Committee, makes decisions regarding the validity of GM research and the safety of introducing GMO for public services. Developing countries are rich in biodiversity and traditional knowledge related to bioresources. Biopiracy refers to the use of bioresources by multinational companies and other organizations without proper authorization from the countries and people concerned without compensatory payment. Controversies regarding patents and biopiracy: 1. Basmati rice. 200,000 varieties of rice in India. 27 documented varieties of basmati rice in India. Black circle in 1997. An American company got patent rights on basmati rice through the U.S. Patent and Trademark Office. Black circle basmati rice, semi-dwarf variety of rice. New variety of basmati rice, turmeric neem. The Indian Parliament has recently cleared the Second Amendment of the Indian Patents Bill.